Periodontal Vaccine by Giovanna, Leticia, and Christy.
To begin, let's review the pathogenesis of periodontal disease so we can understand the strategy behind the development of a periodontal vaccine. As we know, in a healthy periodontium, plaque contains mostly beneficial bacteria that are in homeostasis within the host. But there's a change in the composition of the plaque in patients with poor oral hygiene or certain health conditions. This allows certain bacteria to establish themselves in the tissues surrounding the teeth, or the periodontium. P. gingivalis is the key to this chronic disease because it causes the host's immune system to become self-destructive, and it allows for the emergence of other pathogens such as T. denticola and T. forsythia, known as the red complex. P. gingivalis is needed in order for the other accessory pathogens to become established in the periodontium. Once they are established, the pathogens reinforce P. gingivalis, contributing to chronic inflammation we know as periodontitis. The University of Melbourne, located in Australia, is in the process of developing a periodontal vaccine that targets P. gingivalis. The vaccine creates a switch in their immune response from self-destruction to protection, and it encourages the production of antibodies. The antibodies then act to block the red complex's ability to cause disease and allows the beneficial bacteria in the plaque to outcompete the disease-associated bacteria, which brings back homeostasis again within the host. The inflammation subsides and the progression of the tissue destruction ceases. So far, the research published from Nature partnered journals has only tested the vaccine in mice. Once trials are proven successful in humans, the vaccine will be able to prevent the main cause of tooth loss in people over the age of 30 years old globally. You guessed it, periodontal disease.
Studies show that a periodontal disease vaccine can become an important part in preventing the recolonization of P. gingivalis and can reduce the progression of disease if and when used in addition to mechanical debridement, such as scaling and root planing. Immunization against P. gingivalis can reduce periodontal bone loss at mucosal sites by blocking the antigens related to destruction. Antibodies, immunoglobulin A, and G4 in humans can reduce inflammation related to chronic bacterial infections at mucosal sites. Immunoglobulin G4 produces interleukin 10 that has a potent anti-inflammatory property that prevents the damage to the host or person.
Proof of concept. So this was a study conducted in 2019 titled Immunization with Self-Regenerated Vaccine Protects from P. gingivalis-Induced Alveolar Bone Loss. And this study was published in the Journal of Clinical Periodontology, and it aimed to investigate the cell-free protein synthesis, which is basically in vitro, as a means of producing vaccinable targets against P. gingivalis-induced bone loss in mice. 5 groups of mice were separated, each with a different combination of immunizations and oral challenges. Baseline serum was obtained, and then a P. gingivalis slurry was applied to the gingiva of the mice. After 42 days, the serum of the mice were taken again to measure the number of antigens present. Their heads were processed for oral bone loss measurements and then compared to the control group. After evaluation, it was found that the vaccinated mice responded with an increase in molecular-specific IgG antibodies over the control group. It was also found that they were protected against P. gingivalis-induced bone loss. Therefore, a proof of concept was developed.